Sjors Scheres' work goes right to the heart of Sidney Brenner's dictum that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. For the last almost 20 years, Sjors has been working out how to go from the very noisy multiple images of molecules taken in the cryo-electron microscope to highly detailed three-dimensional reconstructions. And his groundbreaking innovations mean the field has reached a milestone in cryo-EM structure determination, the ability to build atomic resolution models of the complex macromolecules of life. Sjors was recruited to the LMB as a group leader in 2010 and is now joint head of structural studies. He was elected to the Royal Society in 2021 and earlier this year was awarded the Royal Society's Van Leeuwenhoek Medal in recognition of his work. Sjors, thank you very much for talking to us today. I'd like to start off with when you first got here. So you were recruited in 2010. What were your first impressions of the place?
Well, I thought the place was very refreshing in that there was lots of very intelligent people together and they all had only one goal in common, which was to do the best possible science they could do. And I'd previously been in other places. I did my postdoc in Spain, and there sometimes I had the impression that that was not the only goal people had, that all kind of other things played a role in how the place was run. And so being here and it was all about your science was a very refreshing take on how to do science.
So the ethos of the Structural Studies division seems to me to be a bit different from the other divisions here, but also from how academic life works generally. It seems much more, as you say, dedicated towards a goal rather than towards the individual egos of the people involved. Would you say that's true?
I think what may be unique about structural studies is that we all use similar techniques to determine the structure. So individual group leaders have their own biological molecules that they're interested in and thereby their own scientific interests. But what binds them all together is that we all use either X-ray crystallography or NMR or cryo-electron microscopy to get to those goals. So that by being bound together to these techniques that we all use, that kind of brings cohesion to the division. We all use the same instruments that we all share, and it's in everybody's interest that we find out how to use those in an optimal way, etc. So I think that's perhaps different from other divisions where group leaders also have their own biological interests, but very often they then use their own rather specific techniques to study those. So I think that might be at the base of what you're referring to.
Yeah. And I imagine that makes it easier to get very useful feedback and critique of your work if people know what you're talking about at a very fundamental level.
Yeah, so when people give talks, it's not only they get feedback that's useful for them on the biology, but also on the actual application of the techniques, which might be far from straightforward. So there will be experts on that technique in the audience very often, and there is a very good level of feedback on how you've applied the techniques.
Yeah. So this
just a biological problem.
This critical mass of structural biologists is really important for the success of the whole division then.
And the whole LMB, I would say, as structure seeps out into all the other divisions. Again, I think highlighting how important it is how informative structures are if you want to understand how proteins work.
Yeah. Do you think there's a particular sort of scientist that's attracted to the LMB?
I don't know. I think scientists at the LMB are encouraged to work on important problems, on difficult problems that go perhaps beyond the difficulty of problems that you could be dealing with in other settings, in universities or so. And so I guess the scientists here need to be, in that extent, somewhat more courageous, or kind of, you have to be willing to get your teeth into a very hard multi-year project that perhaps the outcomes of it are less clear. But I think ultimately scientists in many different settings would have much more in common than what scientists at the LMB have different from all these other settings. So I think we have more in common than what divides us, I think.
Do you think there's a pressure on people here to come up with brilliant things then, rather than otherwise?
I guess people have to come up with good science everywhere. So, but yes, there is definitely some pressure on people to, especially, yes, I think so.
Yeah, I mean, when you've come into a lab with the number of Nobel laureates that a place like this has, it must place a certain pressure on new recruits, you'd think.
Yeah, I think many people compare themselves to other people, and how am I doing compared to my colleague X, Y, and Z, and that's in itself is not so useful, I think, because it often leads to anxiety and negative emotions, etc. I think I would recommend people who come, new recruits, to focus on the science that I'm doing. Is that important science? If it all goes well, will it answer questions that people are really interested in, that will change things as we do things, or change how we understand the world, rather than compare yourself to how other people are doing. I think that's often a recipe for unhappiness.
It's very difficult not to do that, given the current systems of evaluating people, I guess, by funding bodies and so on. But
I guess more than funding bodies and the system you're in, it's probably more a human characteristic of human nature that you tend to compare yourself to others. But I think it's probably wiser to consider in a more objective thing that what you're doing is the right thing. And if things are not going well, you go back to your first question: is it worth doing what I'm doing? Yeah. If things go wrong for a prolonged period of time, perhaps what you thought was a good idea is not a good idea, and you should abandon it. But there's quite a lot of time that if you think you are doing the right thing and it should be possible, then you should stick to it and keep going at it, I think. Yeah.
And you're now joint head of division of structural studies. Was it what you expected it was going to be when you took the job on?
Yes, I think so. It's taking a bit more time than I had expected. Sometimes it comes in waves, so sometimes there's a lot of little things you need to do, and other times there's prolonged periods of, weeks that it's relatively quiet and you can really focus on your own research in your own group, etc. Yeah. So, but I do this, I think what is good is in structural studies and several other divisions, we have 2 heads of division. So I really enjoy doing this together with David Barfoot. And, I've learned a lot from David doing this now for almost 4 years, I think. So, and I enjoy going up to David and having a little chat almost every day, to be honest, on the things that are going on and what does he think, and we bounce a few things back and forth. And that, I find, is then, it takes a bit of time to walk up and down and to have that chat, but it's pleasant, and I think it's overall quite an efficient way of making good decisions, because if you would have to think about all the decisions yourself, then you just sit there thinking and there is no kind of echoing the idea around. So it's enjoyable and efficient, I think. Yeah.
And it's really nice, I imagine, having someone who's on the same sort of level as you, so that you can each tell the other person quite frankly what you think, rather than...
I try to do that to whoever person I speak to. So.
I imagine one of the pleasanter things about being head of division is recruiting new people. So what is it you say to new recruits to hook them into the LMB?
Oh, I tell them how great it is not to be overwhelmed by all this administrative things that they would have to do in other places, right? I tell them you will not need to write all those grants, for example, especially if you want to, for young group the program leader tracks who come, I would say I would encourage you not to apply to Wellcome Trust or EU type of big grant type of things, because you get core funding from the MRC with your 2 postdoc posts and your 1 or 2 students, so you can grow to a certain size where you can have a good, effective team where you still very much involved yourself in the research. Many people who've done a postdoc and become a group leader somewhere, they then complain that they would like to be a postdoc again because they can't do the science themselves anymore. So I tell new recruits, here you can still be a postdoc. You can work as a team with your postdocs and your students. And, if a few years down the line you want to grow that enterprise and be bigger, etc., you can still write grants. But for the moment, when you start, forget about all those things and just focus on getting your science done.
Yeah, and I imagine that's quite attractive to potential recruits.
Yes.
Yeah. Is there something else that particularly enthuses people about it, that makes them want to come here?
I think the group of people that are here, structural biologists coming into the structural divisions at the LMB, I think they are very, very keen to work with all the people that are here whose names they know and they value from conferences where they've coincided, etc. So, and we are a very collaborative division. We mentioned about the methods binding us all together. So interacting in that sphere really helps people along, and it really accelerates people's science when they come here compared to a place where there would be much less of those interactions. If people give a talk on a structure they're trying to solve in our weekly colloquium series and they say, I have these and these problems, there might very well be people in the audience who say, but why don't you try this and this? Or, people meet each other over a coffee and then, so that type of feedback then leads to much faster science for many people compared to if they're in a more isolated environment. So I think...
Sounds perfect.
And then another thing, especially for structural biologists, we have a good amount of electron microscopes, for example, here. And so the amount of infrastructure that you would perhaps need to set up yourself or kind of hitchhike onto something that somebody else has set up in the university you're going to, compared to what they would have access to here, is also very appealing, I think, for many people.
Yeah. No, I'm sure. So that sort of brings me to your beginnings here, which also brings us to this keyboard that you brought with you. So this is the first keyboard you were given in 2010 when you started here, is that right?
That's correct, yes.
Tell me all about it.
So I thought I'd bring this because for me this symbolizes what the pipette means for the typical molecular biologist. So it's your main tool to do your own science. And I came here to work on image processing for cryo-electron microscopy. And so my background, my postdoc was in image processing development for cryo-EM, and my PhD was also on methods development for X-ray crystallography, structure determination of proteins. So my expertise does not lie in the wet lab. I'm not very good in the wet lab. My expertise lies in computer programming and designing computer algorithms to deal with the type of problems that we have in cryo-electron structure determination. So I thought that rather than bringing a pipette, I would bring my keyboard.
Fair enough. So you said you started off as a X-ray crystallographer rather than a cryo-EM person.
I did.
So did you do wet lab stuff then?
Yes, in my PhD, I did my PhD at Utrecht University in the Bijvoet Center, and I started off doing the typical structural biology project of using X-ray crystallography, where I expressed a protein. It was a transcription cofactor, and I tried to grow crystals of it and then bring them to the synchrotron and solve the structure. And that was a frustrating endeavor for me. So I did not like the wet lab work where you had to make your protein, express it in E. coli, all smelly, and then disappear into the cold room for 2 days to run all kind of columns, and then run a gel on Friday evening to find out that the protein had broken down again, etc. So I did solve a few structures, and I found out during the first 2.5 years of my PhD that I really enjoyed taking X-ray diffraction data and processing X-ray diffraction data and thinking about how that all worked. I loved the predictability of X-ray diffraction pattern and how you could use math to describe it all very well. But I did not like the actual wet lab work, and I wasn't very good at it. So after 2.5 years, when there was no structure, we decided, my supervisor Piet Gros and I decided it would be a good moment to change direction, and I then changed to a methods development project on which in the end I wrote my PhD thesis on after ending my PhD in 5 years' time, which was still kind of a normal time in the Netherlands.
And you've said that you needed a younger field than X-ray crystallography.
Yes. So when I did my PhD in X-ray crystallography, people already were. It was all about structural genomics and automation. So people were building robots for the beamlines, and people were speaking about automated data processing, etc. And I felt that the field was too mature. That if you could probably have fun automating things, but in terms of doing new science, I thought that was not exciting enough. So I then wrote to several labs, and the one I ended up going to was in Spain, in a lab, in the lab of José María Carazo, who did image processing development for electron microscopy. And that's where I then learned how to do image processing, and I learned about all the electron microscopy things around it.
What is it you find so fascinating about this then?
So in electron microscopy, we take all these images of particles which are suspended in a thin layer of vitreous ice, and the images, if you look at the raw images that you take on the detector in the microscope, they look more or less like a badly tuned TV. You can hardly see anything in it, so they're extremely noisy. Yet hidden in that noise is information about the atomic structure of these proteins. So I find the problem, it's a mathematical problem of how to go from these very noisy images of particles which you don't know which orientation they were in when you made that thin layer of ice, and you don't know exactly where they are, and often these particles adopt multiple different three-dimensional states because proteins are little machines and they use motions of individual parts as an intrinsic part of their functioning, and all of that information is in the images, but you need to extract it out. So I'm intrigued by the mathematical problem of how to do that.
And do you like the idea of establishing order?
I do like order, yes. I think most scientists like order.
I guess so. And your big insight into how to do this came from your experience in X-ray crystallography, is that right?
Yeah, so one aspect of how to do this had to do with what do you do with parts of the information that you need to get to the structure that was lost in the experiment. And X-ray crystallography, you measure the intensity of diffraction spots, and the phases of each reflection are lost in the experiment. You only have information about the amplitudes through the intensity of the spot. And in X-ray crystallography, the way to deal with that, they call it a missing data problem, is something called marginalization, where you come up with a statistical model and then you calculate probabilities for all the possible phases and you integrate over those. And it was while I was in Madrid with José María Carazo that I started reading up on, what does that mean for electron microscopy? In electron microscopy, the images are taken with a microscope and they contain both amplitude and phase information. So that it was not a phase problem like in X-ray crystallography. But what is lost in single particle cryo-EM is the actual orientation of that particle. in what way was it tumbling around when the picture was taken? Is it a top view or is it a side view? So it was the insight of you'd have to marginalize over orientations rather than over faces that I was very interested in. And I found a paper written by Fred Sickworth in '98 that did this for a very simple toy problem of 2D refinement with a single reference. And it was that kind of concept there that I then expanded into multiple different 2D images where you have top views and side views, etc., all together, which you want to separate, and ultimately into real three-dimensional classification where you have projections of images in all different 3D orientations and multiple structural states, and you all separate it out together. And marginalization there was very important. And then when I moved to the LMB, I built on that. So I then made another very important, I got a very important insight in that regularization. That's kind of how do you prevent noise to build up in the structure that you're calculating, and that you could do that in a Bayesian framework. So the insight there was on how to come up with a explicit mathematical optimization function that would lead you to the least noisy reconstruction that you could get given the amount of data and that you have at hand.
So this wasn't something that had been done by other people before then, apart from that one paper that you alluded to?
Yes, so the marginalization had been done only for alignment of a single 2D toy problem. So there, the extension to multiple classes and ultimately even doing it in 3D was my contribution. And the regularization, nobody had really written out how you would explicitly do that. People had done all kind of ad hoc solutions, and some of them were relatively close to what you could get to in this Bayesian framework. But the realization that you could it all write down as one optimization target and how you would have to optimize that was work done here.
What do you think it was that gave you your edge in this then? Why were you the person who could make this insight?
Oh, I don't know. I think in Madrid, so marginalization was definitely through my background in X-ray crystallography. So I think changing fields is very refreshing because you take your expertise from one field and you can apply it to a new field where people who've been in that field for a while may not have thought about it that much. Okay, so I think, and then the regularization, I think by then had been thinking about this problem for many, many years and you know, banged my head against the wall in multiple different ways. So that then it was actually Gary Murshadov, another group leader in the structural studies division, who's a mathematician. He said, "Oh, you should read this book on Bayesian statistics by Rob McKay here from Cambridge University." So he gave me the book, which he had in his office. I read the book, and then I think because I had all that knowledge in my head, it all kind of fell together, and I had a aha moment. I said, "Oh, that's how you have to do it."
That must have been really exciting.
It was exciting, yes.
So does that mean you retired to a darkened room for some days with your keyboard to type frantically, or what do you do when you have that sort of mathematical insight?
Well, I had already kind of retreated to my room for quite a while. So when I arrived to the LMB in 2010, I didn't hire any group members until early 2012. So for about 2 years, I was programming on my own. So but when I had this insight, yes, then you go and try and implement it. And it still takes, it's not something that you then just write up and do and the next day it's there. It's still a process of many months before it all really clicked all together as it should do.
But it was obvious to you from the beginning that it would work. You just had to...
Once I read that book, I was convinced that this was the way to go, yes. And I was then also committed to spending that time. That comes back to this early point we touched on. If you think you have a good idea that's worth it, then you should do all you can to make that idea work.
So were you worried that other people had the same idea?
No, not really, no. I try not to worry much.
That seems like a good philosophy. And you've said that you were at the right time in the right place working on the right thing. So why was it so important to be here? Because you were in that cluster of people who understood the problems very well. And
Before in Spain?
No, here, I mean.
Oh, here, yes. Yeah, so why was I at the right place at the right time, doing the right thing, is because the reason why Richard had kind of recruited me was because he knew that his developments on new electron detectors were going to come to fruition sometime soon, and he expected they would make a big difference and move the field forward by a large step. So they wanted somebody to do image processing here at the LMB, which in the end turned out to be myself. So then when RELION, which is the name of the program that we implement all these developments in, when that became fully functional, that was around 2012. So that was when I started hiring my first people in the group. And then, so Sjors Bai was my first postdoc. He then started working with Israel Fernandez, who was a postdoc with Venki Ramakrishnan's group. And so Israel would make ribosomes and Sjors would image them on the new detectors that we then had a prototype of that had been installed on our Polara microscope. So there was very good samples from the Ramakrishnan lab, my postdoc who knew very well how to take good images, and then we had my software to process the images. And then suddenly we found out that you could go to resolutions that were good enough to do de novo atomic modeling in these cryo-EM reconstructions, which, that was the first time we saw that. So that was very exciting. Yeah. It's definitely various things coming together.
So how much of a jump in resolution did all that achieve then?
Oh, so before then, I would say there was some icosahedral viruses, very large viruses where you have multiple copies of the same protein that with a defined symmetry among them. For those type of objects, close to resolutions enough for de novo atomic modeling had been achieved, around 4 angstroms, say. But for anything with lower or no symmetry, that was definitely not achieved at all. You would get stuck at resolutions which, if you were lucky, you kind of saw individual alpha helices as tubular densities in the maps, say around 8 or 9 angstroms or so. It was suddenly going from that type of maps to maps where you could see the entire main chain and many of the side chains, and you could think of doing de novo atomic modeling. That was kind of really the jump. So we went from, later people have called it blobology before, where you had kind of blobs for protein domains, to maps with sufficient detail to do atomic modeling.
So that's an extraordinarily big jump then for the whole field.
Yes, so that's then has been termed the resolution revolution for cryo-EM, etc. Yes, so and that then has led to a major influx of people to the field, and nowadays it's routine to do structures to 3 angstroms or even 2 angstroms now. So,
what sort of people do you have in your lab now then?
Oh, so half my lab works still on algorithm development that we implement in RELION, and they are physicists or computer scientists or mathematicians. And the other half of my lab works in a collaboration with Michel Goedert, where we then apply the methods that we make to a specific biological topic. And in this case, that's amyloids.
Yes, I want to talk about that in a minute, definitely. So your software's RELION, is that's yes, right? That's correct, yes. And you're very passionate about it being all open source and available to everybody.
Yes.
Yeah. Where did that come from?
Right. So I think part of it comes from my PhD already. So my PhD supervisor, Piet Gros, was involved in software development, and he was involved in the development of Explore and CNS Solve. And there had been major problems in the X-ray crystallography field there because of certain licenses, etc., and companies running away with software, etc. So I think I got kind of a background of how important it is to have open software during my PhD already. Then when I came to Carrasco's lab in Madrid, they did everything with a very open source type license, a GNU public license. And I kind of learned that their software already was built partially on software that was developed in Joachim Frank's lab, the Spider software. And that's where Jose Maria did his postdoc. So you could see how by keeping software open, you get very rapid progress of science, because the next people don't have to invent everything from the ground up. They can build on things that other people have already done and looked how they did it, and thereby progress their science much faster. So when I came to the LMB and I decided to write my own software package, which is now RELION, I also used pieces of code from the software from Jose Maria Carazo, XMIP, as part of the code base of RELION, because that's what you're allowed to do, and it makes that you don't have to build everything from scratch. So, and I also looked at open source code from Nico Grigoriev, who had worked previously here with Richard Henderson and did reconstructions in a particular way that I liked. And I looked at software from Bernard Hyman, who had written certain libraries which I really liked. So by taking the great from many smart people, you can make your own software progress, your developments much faster. So I think in general that leads me to a conclusion that open software accelerates science, and that's what I like to rant about on Twitter.
How did you come up with the name for it?
RELION? Yes, RELION stands for regularized likelihood optimization. So it's yes, but the kind of the joke was that it would be software you could rely on.
Yeah, indeed.
And that's why you pronounce it RELION rather than RELION or something like that. So, and that was because this Bayesian framework learns how to optimally get the least noisy reconstructions from the data themselves. So that's characteristics of many of these Bayesian approaches, that they learn automatically or kind of from the data how to do things in an optimal way. Whereas previously you'd need an expert user to tune all kind of parameters to approach the optimum, but you'd have to be an expert in doing so, and you'd have to. So that's where they kind of rely on the new software came from.
Are there elegant ways of writing this sort of program and horribly ugly ways? And does that matter?
Yes. And, I wrote RELION with what I knew back in 2010, and I started here, and I've learned new things ever since, of course. But yes, it does matter, especially how long your software can hold up in the future and how easy it is for other people to make contributions, etc. So now looking back, now it's 2022, and I've realized, certain decisions I made were not the best ones. If I have to rewrite it again, I would probably do some things differently. But also, the way you write computer programs is progressing as well, just as our science, right? So with modern insights and modern languages, etc., you could do things differently now. Overall, I'm not too unhappy because I think external groups have made very useful contribution. Eric Lindahl, a collaborator of us, did acceleration of our code on graphical processing units or GPUs. And so his students dove into my code and got to work with it in a very efficient way. So I think, and other people have also built other things on it. So overall, I'm not too unhappy, but there's always things that if you look back 10 years, you think, oh, I wished I had done that differently. Yes.
So you don't feel proprietorial about RELION then?
No, anybody can copy it and change it as they like. Yes. Yeah. So I think that's the key of open software, that you should let go that this is your thing, and to some extent it is my thing, it is my baby. But if somebody takes my baby and does something great with it, then I should rejoice, because that means that I've had an impact on their science and on science in general. So this is not something that you should try to restrict. You should actually try to actively promote.
Where do you see RELION going in the future then?
Oh, I don't know. I hope it will continue to to assist people in their structured determinations for many years to come. At some point, we'll need a rewrite at some more basic level, I think, because as things get to be done in more modern ways, and there's different ways you can accelerate code, etc., then you need to, it's not something that's done and then lives forever. You need to, if you want to stay relevant, you have to keep working on it all the time.
Sure. And that's something you plan to carry on with then?
Yes.
Yeah. So just turning to the other side of your lab, so you collaborate very closely with Michel Goedert. Can you tell me something about that collaboration, starting with how it came about, I guess?
Right. It started when Anthony Fitzpatrick became a postdoc in my lab. He'd gone away on a Marie Curie fellowship, I think, to the US. He'd previously been in the Dobson lab here in town at the chemistry department. And he had done cryo-EM of amyloid filaments together with Helen Sable, also at Birkbeck College in London. But then he'd gone for a postdoc 2-year stint, I think, in California with the whale. And then he kind of realized that was not the direction he wanted to go in for his career. So he had to come back for 1 year on this Marie Curie fellowship. That was a condition of the fellowship. So he then wrote, I think originally to Richard, say, can I do this 1 year in your lab and learn single particle cryo-EM, because he thought that would be a more fruitful way forward in his career. So then Anthony came, and then instead of Richard said, why don't you take him on? Because Richard wasn't so keen on taking on new people anymore. So I then worked with Anthony. He learned how to do single particle reconstruction in my lab. And then at some point he gave a lecture in our colloquium series and presented his previous work that he'd done with Helen Sable. And then Michel, Goedert, and Tony Crowther came to the lecture. And after that I said, perhaps we should, with the whole resolution revolution and RELION, etc., we could go back to it. And that more or less coincided with a PhD student in my lab who'd implemented helical symmetry inside RELION, because these are helical filaments and they need processing in a specific way. But that software was more or less ready. So we then formed a team with Anthony and Sjors and myself from my group, and then Ben Falcon and Michel from the neurobiology side. And Ben extracted filaments of the protein tau from the brain of an Alzheimer patient. And then Anthony imaged those filaments, and then Anthony, Sjors, and myself together, we kind of figured out on how to solve structures of these amyloid filaments, which nobody had done to kind of resolutions again enough for de novo atomic modeling. But after a while, we figured out how to do this, and then got to this first structure of how tau filaments look like in the brain of Alzheimer's disease patients.
And what has that done to the Alzheimer's field then, having this ability?
Right, so it has taught us which part of the protein are forming these helical filaments and which parts remain unordered. And I think, but most importantly, I think there's the same protein tau goes wrong in many different neurodegenerative diseases. And Alzheimer's disease is the most common one, but there's a whole series of around 20 or so different diseases which are commonly called tauopathies, because they're all characterized by tau forming filaments. And Anthony then left. He became assistant professor in Columbia University, and then we continued to work here with Michel and I. And since then, we've done tau structures from almost all of these tauopathies. But there's still a few rare ones pending, but we've done the most important ones. And what we found is that even though this is the same protein, in the different diseases, it folds up in very different ways. And the different structures characterize the different diseases. We've now come up with a structure-based classification of these diseases.
Wow.
And I think that Tony Crowther back in the 90s got some early insights that this might be the case, but we now know at the atomic level how different these structures are. So we can now try to find out why that is. Why is it different whether you have CTE, chronic traumatic encephalopathy, which is the disease that you may get if you get repeated head traumas. There's some famous cases in the US for American football players, for example, or boxers. Yeah. So why does those structures look different from the one in Alzheimer's disease? We don't know, but I think being able to find out it will be very important to understand these diseases.
And also how a protein can be so malleable in the way it folds, presumably.
Yes, it's very different from from normal globular protein folds, where one protein sequence through evolution has kind of come to a solution where one protein sequence adopts one globular fold, or with a few possibly different conformations in the functional cycle. But amyloids are very different. They're probably not functional in the sense that, you know, they're beneficial for your survival and play a role in evolution. So, but somehow there is a lot of malleability, as you said. So the same sequence can form multiple different amyloids. And we have some ideas of why that could be, but how it happens specifically for these different structures in the different diseases, we have no idea yet. And I think it's important to find that out.
So you're as fascinated by this biological problem as you are with the computational problems then?
Yes, I've taken quite a liking to the problem. So when I came, I first collaborated with Wenke on the ribosomes, and then I collaborated with a Chinese collaborator, Yi Yongxi, on gamma secretase and several other complexes, and then I collaborated with Kiyoshi Nagai on spliceosomes, etc. But all these were structural biologists who basically wanted to learn how to do cryo-EM, structure determination, learn our tricks. So I would help them along and we would work together for a while, but once they would be independent, they didn't need me anymore, they would move on, which suited me fine. I think that was, you know, the ultimate success of the collaboration. And while we were collaborating, I would learn a lot about their types of problems and design specific solutions for them. So this was mutually beneficial. But the great thing with Michel is that Michel is a neuroscientist, so I can not only be the cryo-EM trickster, I can also be the structural biologist. So I've now, and that's where this collaboration then becomes much more long-term and also more intellectually stimulating, where I can really dive into a biological problem and, you know, become excited about it. And, you know, I now, as a structural biologist, I just find it fascinating how this one sequence can form all these different structures. Besides the obvious, you know, interest for understanding neurodegeneration, already from a structural biology point of view, this is kind of amazing, and I want to understand how it works.
Yeah. And the key to the success of this is that you're both very different sorts of scientists then.
Yeah, I think our collaboration works really well because Michel brings in all his expertise about neuroscience, and I bring in the expertise about cryo-EM and about, you know, protein structure in general. So I think often the key to good collaborations is that the different collaborators are very good at different things, so they complement each other well. I think those are often the collaborations that work out most excitingly, because when I was in Madrid, we had many collaborations with people who did very similar things that we did. They also did image processing and they worked on similar problems, and then, let's work together. Yeah. But I've never found that to be so stimulating, because then you all work on the same problem just because you kind of have similar expertises. Then why do you really work together other than kind of feel warm and fuzzy about it? So I think it's much more exciting if you work with people who can do things that you have no idea how to do.
There's a certain degree of trust involved in that as well, though, isn't there? That the other person is as good as you are, almost.
Yes, of course. But there is whenever any type of collaboration, when you start a collaboration, it's based on just trust, I guess. You can inform yourself, but ultimately these are all people, and you're going to work together, and you're going to trust that the others are going to behave in a certain way and going to contribute certain things. So I think trust in a collaboration is always very important.
So what is it that excites you most about your work these days?
I get excited about many things. So I get excited about sitting with my students or my postdocs and go through their latest data, be it algorithm developments and, you know, what you can learn there to do image classification of ever more difficult mixtures, you know, whether that suddenly there is a data set that we couldn't handle, and then there's a new algorithm and the postdoc says, look what I found, and I found these things that I can now separate, etc. And the map looks much better than with anything else. And that's great, because then, the biologist who is involved can go on and do their biology, understand their biology better, etc. So, but on the other hand, I also very much excited about the work that goes on with the amyloid. So I think I'm very excited, for example, Sophia Löwestam, a PhD student in the group, who's jointly supervised between myself and Michel. So she's been able to make the specific structures that tau adopts both in Alzheimer's disease and in CTE in the test tube. So as before, we could only get those structures by isolating them from brain. And if we would try to make them from recombinant protein in the lab, then those structures would look different. And Sophia's found out how you can make the exact same structures. And by doing so, we now have a way in the lab to make them. And that means we have very pure preps with a well-characterized structure. We could start to do kind of comparative studies, how if I put these on a certain model system of disease, how does it compare the AG structure compared to the CTE structure, for example, compared to the structures that were not relevant in disease? And you could do comparative experiments in trying to understand what would happen in disease ultimately.
Yeah. So you're not done by a long stretch yet in either side of your interests then?
I hope not.
Thank you so much for talking to us. That was really interesting.
Thank you, Kathy. Those were great questions.